So I'm going to tell you about a couple of projects from my group. And my group, we are interested, as are many groups in genetics, in understanding this question of how does variation at the DNA level impact human phenotypic variation. And we, of course, think this must happen through cellular and molecular mediators. And one of the molecular mediators we're particularly interested in is RNA splicing. So as you probably know, splicing is the process by which these non-coding intronic regions are removed from the initial RNA that's transcribed. There can be variation in what regions are removed as intronic, which gives you different splice isoforms, and that can impact phenotypes like disease risk. So I'm going to tell you about a couple of projects sort of spanning this diagram. The first is going to be trying to get a better understanding of how genetic variants can affect splicing by looking at genetic effects on splice factor binding. I'm probably not going to have time to tell you about work we've been doing on splicing modules, so this is trying to look at transcriptome-wide patterns of splicing variation. But I will tell you about stuff we've been doing on causal network inference to try to connect together intermediate phenotypes like gene expression and splicing and traits like disease. So the reason I initially kind of got really interested in splicing was through co-developing this method called leafcutter during my postdoc. So this is a flexible, scalable way of quantifying splicing from short-read RNA sequencing data. The basic idea is illustrated here. So if you have some gene here where the black blocks are corresponding to constitutive exons and the white to alternative exons or sub-exons, then what leafcutter does is to look at the spliced reads in this region from your RNA sequencing data. So these are reads where part of the read aligns to one exon and the remainder to another. And what the algorithm does is to construct this intron graph. So here, nodes correspond to starts and ends of these splice junctions, or introns, if you want to think about it like that. And the edges are the junctions themselves. And then we look for connected components in this graph. So a connected component is just a set of nodes where you can get from any node in that set to any other node by traversing edges in the graph. Then the quantification of splicing that we use is to look at the number of these spliced reads that correspond to each edge relative to the other edges in the same connected component. We have some statistical machinery based on a Dirichlet multinomial generalized linear model that lets you test the differences in splicing between different conditions or between different genotypes. One thing you can do with leafcutter is to map splicing QTLs. So we've heard a bit about expression QTL. So this is the analogous concept, but looking at splicing variation instead of expression variation. And GTEx have done this, for example, mapping splicing QTLs alongside their expression QTLs in many different tissues. And it's been seen repeatedly now, we've done these analyses as have others, that splicing QTLs are often just as enriched and sometimes more enriched in GWAS than eQTLs. And so it depends on which trait you're looking at, which way this goes, but the big picture is you want to think about splicing as well as expression when you try and understand what effects your GWAS variants are having. But how do SQTLs actually affect splicing? So I took this figure from the most recent GTEx paper, where they looked at enrichment of eQTLs and SQTLs in different genomic regions. And some of the things pop out here that you might expect. You're more likely to be an eQTL if the variant is in a promoter. You see some enrichment for transcription factor binding sites as well for eQTLs. And for SQTLs, variants that are at the splice donor or the splice acceptor, so the two ends of the intron, or in other known splice regions, are more likely to affect splicing. But one thing that's noticeably absent from this figure is anything about RNA binding proteins or splice factors. And that's really surprising because our understanding of how splicing is regulated is that that's done through a combination of cis-acting RNA sequences in the actual messenger RNA itself, and then these trans-acting RNA binding proteins known as splice factors. So this is a figure I made at some point after doing my best to read a molecular biology textbook that went through all the detail of how splicing works. You can see there's a lot of proteins and non-coding RNAs involved, and this is only a subset of all the proteins that are known to be involved in splicing. But the important bits for the point of view what I'm going to be talking about are these things like exonic splicing enhancers and exonic splicing silencers. So these are things where proteins can bind to the RNA and influence whether this region is going to get included in the final mature messenger RNA or not. Those proteins include things like SR proteins and HNRMPs. So the biggest resource that we have for looking at where do RBPs bind in cells is from ENCODE. So they did ECLIP, which is sort of like the ChIP-seq of the RNA world, to look at where do RBPs bind in human cells. And they did this for a really large number of RBPs, over 100, in 2 different cell lines, so K562, which is an erythroid progenitor cell originally, and then HepG2, which is from a liver cancer. This is just showing the number of peaks that they find for different RBPs. It's quite a big range, but this is kind of the gold standard data set at the moment. So the first thing we wanted to look at is, are splicing QTLs enriched in those parts of RNAs that are bound by RBPs according to ECLIP? So here, my postdoc Megan Schertzer took those ECLIP binding peaks from ENCODE for HepG2, which is sort of liver-ish, and cross-referenced that with leafcutter splicing QTLs from GTEx for liver tissue. And she found across the majority of RBPs, she found these quite strong enrichments that indicate that a variant is more likely to be a splicing QTL if it falls into an RBP binding peak. But of course, just because a SNP falls in a peak doesn't mean it's going to change binding of that RBP. And there's really been very little work trying to figure out which variants actually might affect RBP binding, with the notable exception of this paper from Grace Zal's group, where they looked at the ENCODE data, and they looked at heterozygote sites in these 2 cell lines, HepG2 and K562. They counted how many of the ECLIP reads were coming from the alternate allele versus the reference allele, and then came up with a statistical test to ask, was there allele-specific binding? So you're seeing more binding of the RBP to one allele versus the other. And they were able to find hundreds of SNPs where this seems to be the case. But this is limited, of course, because you can only look at SNPs that are heterozygote in these 2 cell lines. So since I did my postdoc at Stanford, I was very aware of this work from Hunter Fraser's group, where they had pooled individuals and then done individual cell lines, and then done ChIP-seq, and they also have follow-up work where they do ATAC-seq and look at DNA methylation. So the idea here is, by doing these pooled experiments, you can look at much more genetic variation than if you were looking at a single cell line, but you also get to remove a lot of the technical variation that comes from separately profiling multiple individuals. And especially for things like transcription factor binding or splice factor binding, we expect that most of the time the effects of variants will be relatively local. So looking at allele-specific patterns is very powerful. One of the other reasons, apart from controlling technical variation across individuals, that the pooling approach is appealing is you actually need a surprisingly small number of individuals to capture a lot of common genetic variation. So this plot is based on data from GTEx. So if you pool just 10 individuals, you already represent around 95% of common SNPs, where I'm defining common as higher than 5% minor allele frequency. The data I'm going to be showing you is at this point on this graph with 9 individuals pooled. So what Megan did in these experiments was to take 9 iPSCs, so those are induced pluripotent stem cells, pooled them together, grew them for a while, and then did RNA immunoprecipitation and sequencing. So this is sort of a related method to ECLIP. The real reason we used it was Megan had generated really beautiful RIP-seq data during her postdoc, and so we knew the protocol very well. We also think it does have some advantages. So here I'm showing data for a splice factor called HNRMPK. So these first 2 cyan tracks are RIP-seq data, and there's a binding peak here. And this is the corresponding ECLIP data. And there is a corresponding peak So two things to notice. One is our peaks are generally broader than in the eclipse data, but also we seem to have higher specificity. So you see more of this non-specific sort of noise outside of the peak regions in the eclipse than in our RIPSEQ data. And actually for us, these broader peaks are in a way an advantage because we have more chance of capturing a SNP that's nearby this peak with those broader peaks than these very sort of punctate peaks in the eclipse data. So the idea is after doing the RIPSEQ, we'll then try and identify allele-specific binding by looking at the number of reads that are coming from the reference versus the alternate allele for each SNP. One cute thing you can do with this data as well is to actually, from the sequencing data itself, you can get an estimate of the proportion of each individual that's represented in your pool. So the way this works is by fitting this linear regression where y here is going to be the allelic ratio, so the proportion of alternate versus reference allele at SNP I in your sequencing data. Then x here is the dosage, the genotype dosage of individual j, coded in a slightly unusual way for GWAS people as 0.51. So 0 here would be homozygous reference, 0.5 would be if you're heterozygote, and then 1 would be if you're homozygote alternate allele. Once you fit this model, these beta regression coefficients are going to end up being the proportion of the pool corresponding to individual j. So we can do this for the data that we have. We have two biological replicates, and these, each row here corresponds to a different individual cell line. And we've generated data for 3 different splice factors, so HNRMP-A1, K, and RBFOX2. And then we also generate input as a part of this. So this is effectively just a total RNA sequencing run. And these IgG controls, which are just to check for non-specific binding, but are not going to be too important for the rest of what I'm going to talk about. So you can see the estimates you get are quite consistent regardless of which IP we're looking at. And we sort of had an accidental built-in control here, which is this cell line wasn't growing very well, so we actually didn't include it in biological replicate 1. So the true estimate here should be 0. You can see this is pretty close to 0. We actually have an improved version of this model now where we do constrain the regression coefficients to be greater than, or to be non-negative, because you can't contribute a negative amount of the cell line. But this gives you some sense of the estimation error. Whenever you fit a linear regression, or any model perhaps, it's a really good idea to look at the fitted versus the predicted values, and versus the observed values, sorry. And so we did that here and found this slightly strange behavior. So the x-axis here is the predicted allelic ratio based on those betas and the known genotypes of the cell lines. And the y-axis is what we actually observe in the sequencing data. And you can see it mostly looks pretty reasonable, and then there are all these SNPs where something's getting flipped. And we spent a long time trying to debug this, trying to check whether we were defining the alternate allele differently between the sequencing data and the genotype data. And it turns out it was actually an issue that I was kind of surprised we never heard about. But for Illumina genotyping arrays, and for these iPSCs, we have genotyping arrays, not whole genome sequencing data, unfortunately. It turns out that there can be an issue where Illumina are not actually telling you correctly which strand they are genotyping. And so this is nicely pointed out in this paper, and there's a corresponding script that they provide to correct this if you have the manifest for the genotype file. And after running this, the data looks very reasonable. So yeah, big thanks to them for figuring this out, and good thing to know if you're using genotyping array data at all. And this was work done by my staff scientist, Peter Halmes. One other thing we looked at was to try and understand how much sequencing you need to do in these experiments to have good power to detect these allele-specific binding effects. And so this power analysis, the initial data we had around 65 million reads for the IP and around 50 million for input. And I can go through the details if anyone's interested of the power analysis, we end up estimating you have pretty limited power to detect the sort of effect sizes that we think are there. But if you get up to around 300 million reads in the IP and a sort of comparable number in the input, you start to have pretty good power. So the data I'm going to be showing you is from this point on this graph. The tricky part about trying to say from our data, is there an allele-specific binding effect, is that you don't want to be confounded by there being differences in allele-specific expression. So if one allele is getting transcribed more than the other, for example, because of an EQTL. So we've come up with a Bayesian probabilistic model that tries to explicitly represent both the possibility of allele-specific expression and allele-specific binding. So the way this works is to take those betas, those estimated proportions of each individual in the pool that I told you about, and our genotyping data. And for each individual, that gives us what we think is quite an accurate prediction of the allelic ratio at the DNA level. Then the allelic ratio at the RNA level is going to be similar to that, but it could be a bit different because of allele-specific expression effects. And we model that difference through a beta distribution with a mean equal to this ratio, and then a concentration parameter which controls the width of that beta distribution. So this is the true allelic ratio in the RNA, at the RNA level. So one way of thinking about this would be that it's the allelic ratio you would get if you sequenced infinitely deeply. But the actual observations we have, of course, we unfortunately don't have infinitely deep sequencing. And so what we can use is we use a beta binomial distribution to model the observed read counts, where the mean of that beta binomial is equal to the true allelic ratio, and then there's another dispersion parameter that we're going to estimate. Then the allelic ratio in the IP, again, so that's going to be different from the allelic ratio at the RNA level if there is allele-specific binding of the RBP that we're looking at. So we again model this connection with a beta distribution where the mean is equal to the mean in the RNA, and there's another dispersion parameter, CI. Finally, the data that we have that's informative about the allelic ratio in the IP are the allelic counts from the IP. And so those we model with a beta binomial distribution with a mean of Ri and another dispersion parameter. This is a reasonably complicated probabilistic model. These dispersion parameters are going to be shared across all SNPs genome-wide. And so fitting this is somewhat challenging. And so the approach we've ended up using is to use this probabilistic programming language called Pyro, which builds on top of PyTorch. And the idea of a probabilistic programming language is you can write down a probabilistic model like this in a specific language, and then there is automatic machinery that will do inference of these different parameters and latent variables for you. And Pyro is built on top of PyTorch, one of the big deep learning libraries. So it gets all the advantages of being able to use GPU computation to do all the fitting very efficiently. And the question we're going to be interested in is whether this ratio, so the ratio at the RNA level, is equal to the ratio in the IP. And that's going to be telling us whether there is allele-specific binding or not at each SNP. So for HNRMPK, which is the splice factor that we have the deepest sequencing for currently, we estimate that a bit over 7,000 SNPs show allele-specific binding. And that's around 2% of the SNPs that we were actually able to test based on having enough reads. To contrast this to work that's been done looking at transcription factors, Hunter Fraser's work found that between 1% and 2% of testable SNPs had effects on transcription factor binding if they were in a transcription factor binding peak. So it's sort of on the same order of magnitude. Because what we're really interested in is, do these SNPs that change RBP binding also change splicing outcomes? So the way we're looking at that is to take splicing QTLs from iPSCs. Unfortunately, there's this nice paper published a couple of years ago from Ollie Stiegel and Stephen Montgomery's group, where they did a big meta-analysis of all the functional genomics data from iPSCs that they could get their hands on. So they called leaf-cutter splicing QTLs, and we took that data, we did statistical fine mapping, which gives you these posterior inclusion probabilities, so a posterior probability for each SNP that it affects splicing. And then we use a simple weighted logistic regression model that tries to predict from different features, will a SNP be a splicing QTL? So we put in some control features here, like is the SNP genic? Is it in a gene? Is it exonic? Is it in an exon? Is it in a binding peak for H&R and PK, according to our data? And then finally, is it called as affecting allele-specific binding? So these ASB QTLs. And this is a regression model, so all of these coefficients can combine additively. And so what these having positive significant values means is a SNP that is in an H&RNPK binding peak is indeed more likely to be a splicing QTL, but it's even more likely to be a splicing QTL if it also affects allele-specific binding. So we're really excited about this result. We're extending this currently to more splice factors and also more individuals. So next I'm going to tell you about work we've been doing on causal network inference. And many of you are probably aware of Mendelian randomization since it's being quite widely used in genetics currently. So Mendelian randomization, or MR, tries to get around this issue that if you're trying to estimate the causal effect of some exposure X on some outcome Y, then you're always worried that there might be some confounder U that is associated with both of those and causally influencing both X and Y and making you think there's a causal effect when there isn't. So MR uses SNPs as what are called instrumental variables to try and get around this problem. And it works under two assumptions. One is that the SNPs are not associated with the confounder U, and they only affect the outcome Y through the exposure X and not through any other path. It turns out when you have multiple SNPs that are associated with your exposure, you can actually relax that second assumption a little bit. And that's through an approach called Egger regression, which is illustrated here. So here, every point in this figure is a SNP. The x-axis is the, there should be correlation. This is the correlation between the SNP and the exposure. And the y-axis is the correlation between the SNP and the outcome. What Egger regression does is fits a linear regression through these points. And it turns out that the slope of that linear regression ends up being an estimate of beta, the causal effect. And the intercept ends up being an estimate of the mean of these alphas. So these other paths from the SNP to the outcome. It does not, however, deal with if you have this edge. So if you have an effect going from the SNP to a confounder that affects both X and Y. So the classical MR methods, like Egger regression, consider traits two at a time. So just one exposure and one outcome. And so what my postdoc, Brielin, and I were really interested in thinking about was, could we take some of the ideas of MR and apply them in situations where we have many traits and those traits live in directed networks where they influence each other through the network. And those traits could be biomarkers like gene expression or even splicing. It could be complex traits or diseases. And of course, we're always going to have to keep in the back of our minds that there are going to be unmeasured things as well. So the model we've come up with for doing this, we think is quite simple, but gives a lot of power to try to think about this. So this is a linear model where phenotype J is given by the effects of other phenotypes I through a direct effect network G. And then there are effects of genotypes X via these regression coefficients beta. And then there are going to be unmeasured factors gamma as well. We can put this all in matrix notation like this. And what it turns out you can do under this model is separate out direct effects of traits on each other and mediated effects. So effects where A affects B through some other trait C, or even through longer causal chains. So if we knew the total causal effect matrix R, so this is for every pair of nodes, how much does node A affect node B, including both direct and mediated effects, it turns out that under that model I just described, you could calculate the direct effect matrix G through this expression. So the interesting part here is that is this inversion of the matrix R, and there's some normalization here that's less important. But there's a couple of challenges to applying this expression. I should also mention, for those of you who are aware of Gaussian graphical models or this relationship between if you have a covariance matrix sigma, if you want the thing called the precision matrix, so a covariance matrix tells you about correlations between variables or traits, and its inverse, the precision matrix, gives you what are called partial correlations. So those are correlations between pairs of traits or variables where you've controlled for the effects of all other traits. So the first issue we need to deal with is not all traits are going to be observed. And so this issue sometimes gets called correlated pleiotropy, and the model that Brielin developed to think about this is shown here. So here X would be our genotypes in SNPs and instruments. Then A and B are two traits that we're looking at. And we're going to consider both the possibility that A causally affects B and also that B causally affects A. And we're going to think about there being a confounder U, which itself can have a genetic component. So the approach we came up with to try and deal with this issue of having a genetically controlled confounder works as follows. So the figure here I'm showing you is the same setup as I showed you for Egger regression. So on the x-axis, you're seeing the association between SNPs and A, and on the y-axis between SNPs and B. So SNPs that affect only A are going to be these red points on the x-axis. SNPs that affect only B are these cyan points. And then SNPs that affect U, and therefore indirectly affect A and B, are going to come from these purple, they're going to correspond to these purple points. These are the true effects of the SNPs that we, in practice we have to estimate these, so there's going to be noise on top of these values. And in this setting, this is the null where there's no true causal effect of A on B or B on A. What Brielin's approach does is to calculate these Welch statistics, which have higher absolute value as you move away from the diagonals. And we then re-weight every SNP in the Egger regression according to these absolute Welch statistic values. And it turns out what that does is where Egger regression in this scenario will incorrectly conclude that there's a causal effect of A on B, our Welch-weighted Egger regression does not. And importantly, we still get good power when there is really an effect of A on B, so that's this setting, because the informative points still get significant weights. And so actually here, the estimate from Egger and the Welch-weighted Egger regression end up being very similar. So Brielin did an extremely comprehensive job at looking at when does this work under different genetic architectures, different polygenicity, and different causal effect sizes. And what our broad conclusion is that it's very competitive with existing approaches, but because it's regression-based, it's also very efficient to apply. The competing methods that also perform very well are mixture model-based, so cause and MR mix being the most prominent ones. The downside of these is they're very slow to fit, so if you're trying to apply this to many, many pairs of traits, it's extremely computationally challenging. So there's a second problem we have to deal with, which is this expression I showed you for converting total causal effects into direct causal effects. That was assuming that we knew R perfectly. In practice, we're just going to have an estimate of the total causal effect matrix R. And unfortunately, this is not one of those places where you can just plug in an estimate and everything will work out nicely. And the reason for that is that this matrix inverse is not very numerically stable. So the estimated R matrix might not even be invertible. And even if it's close to not invertible, this can end up being very noisy. The solution we have to this is to, instead of explicitly doing a matrix inverse, we try to find a matrix that's approximately the inverse of R and that has low L1 norm. So that means that its elements are small and many of them are actually 0. So the way we set this up is to minimize this loss function. So this first term is going to encode that we want the inverse to be close to the actual R matrix. And this V is going to be the approximate inverse of R. And this L1 penalty is very analogous to lasso regression or graphical lasso, if you're aware of that. And we set this up in this slightly strange looking way where we actually introduce U as well, and we constrain that the matrix U times V should be equal to I. So that's saying that U is the matrix inverse of V. The reason we do this is it lets us use this very efficient optimization approach called alternating direction method of multipliers. Turns out it's actually a little bit tricky to get this to work, and Brielin had to look up a bunch of tricks to get this to really perform well. But now that it does very efficiently solve this. There's a few things to note about this. So one is we have these weights W, and those are nice to have here because we can incorporate standard errors, which you get from all of the MR methods for each element of R. We have an approach for choosing this regularization parameter, lambda, that's adapted from a similar approach for the graphical lasso. And we're able to use that because it's actually very close in spirit to this approach called the graphical lasso, which is for estimating undirected networks, whereas here we're estimating a directed network. So in terms of how this is going to look overall, we're going to do this pairwise MR estimation of total causal effects of every pair of traits A and B. And then we're going to apply that convex optimization approach to separate out the direct effects from mediated effects to get this direct causal network. Brielin has done an enormous amount of testing out when does this work for different network architectures. So this is interesting in that there's quite a lot of literature on generating random graphs. It often doesn't talk about random directed graphs. So here we want to also think about what's the direction of effect on each edge. So here this is an Erdős-Rényi random graph with just randomly orienting the edges, and there we do quite well. We can also look at hub graphs. So this is where there are nodes that have many connections. Then you need to think about, are those connections all outgoing or all ingoing? This is the setting where the hubs actually have lots of things affecting them, but don't affect that many things. And you can do this the other way around as well. And you can also look at things like scale-free graphs, where there's some nodes with many connections, and then there's a distribution of how many nodes each node is connected to. And in all of these settings, we do pretty well, or very well, relative to the baselines we could come up with. The main place we've applied this so far is to looking at the UK Biobank, which I think you're probably all aware of. So we end up, out of over 9,000 phenotypes that we started with, we filter down to 149 based on them having a reasonable amount of heritability, genetic correlation between male and female, and then also having at least 5 independent GWAS SNPs that we can use for the underlying MR. So we estimate a total causal effect matrix R between all those pairs of 149 traits, and we find around 12% of pairs that have a significant total causal effect, so it's 2,700 causal effects. And out of our convex optimization approach, we get a shrunk or regularized estimate of R. And you can see these actually look quite different. And the reason for this is a lot of these estimates in the original R matrix, although they have high values, high effects, they also have very high standard errors. So those terms tend to get strongly regularized towards 0. And there's around 1,600 entries, sort of substantially large entries in this matrix. So already this is somewhat reduced. And then we can look at the thing we're really interested in, which is this matrix corresponding to the network of direct effects, so having removed mediated effects. And here, this matrix has just over 800 non-zero elements. So what this is saying is those 2,700 total causal effects are explained by just over 800 direct effects and mediated effects. So what's really cool about this is then you can go and look at the characteristics of that directed network, which underlies human genetic trait heritability. So one thing you can look at, for example, is how much mediation there is, and we see a lot of mediation. So one way of looking at that is to ask if there's a causal effect of A on B, how long is the causal chain going from A to B? And it turns out actually the median in our estimated network, the median path length is 2. So that's saying it's actually, if you see an effect of A on B, it's actually more common that that is a mediated effect via a third trait C than a direct effect. Another way of thinking about this is asking what proportion of the variance of the effect of A on B is explained by the shortest path, and that's on average only 52%. So what that's saying is that often when we see a total causal effect of A on B, there's often multiple causal chains by which A is affecting B. We can also look at the in degree and out degree of different nodes. So the in degree would be saying for a given node, how many traits causally affect this node. So for example, you can see BMI has 33 different traits which appear to causally affect it. You can also look at the out degree. So for a given node, how many other traits does it seem to influence? And here you can see hip circumference having a very high out degree of 66, so 66 traits that it affects. To give you a sense of where we're going with this work, we're really excited to try and apply this to looking at gene regulatory networks. So here, this would be attempting to estimate a gene regulatory network that, unlike something like a co-expression network, is directed and also has some causal meaning to it. The data we would need to do this would be well-powered trans, eQTL, and potentially SQTL data. So to do this in collaboration with Tawfiq Raj's group at Mount Sinai, we've been collating and uniformly processing as much postmortem brain RNA sequencing and genotype data as we can to then call trans eQTLs. And we think at the sort of sample size we're able to get to with this, actually based on power analysis based on the eQTL gen data, we should be starting to get to the sample sizes where you can see a reasonable number of trans eQTLs. And of course, because we have this network-based approach that looks at all of the, in this case, genes together, we hope that you maybe don't need quite as large a sample size as you would if you were trying to confidently estimate individual edges. And part of the motivation for this connects to the proposed omnigenic model of complex traits, which says that one of the reasons that human genetic traits are so polygenic is that the gene regulatory networks underlying human biology are so densely connected that you can perturb almost any gene and it'll eventually have a knock-on effect through the network on the trait that you're looking at. Great, so I will wrap up there. Thanks to Lab, especially Megan and Brielin, who drove this work. Thanks to funding sources. We have postdoc positions available, as many people have probably been asking about here. So if you're interested, please do let me know. Yeah, very happy to take questions.